Okay, first of all, I would like to thank the organizer for inviting me and to show you my results and discuss the results with you. Okay, I'm Fabio Mavelli from Bari University. I coordinate a very small group, and our principal line of research is focused on the synthesis of a minimal cell. This is the outlook of this presentation. In principle, we will have just a very short introduction, and then I will show you the result in synthesizing energetically autonomous artificial cell with two different approaches. Okay, and then very brief conclusion at the end. Okay, in principle, this line of research could be located in the synthetic biology. Synthetic biology is an emerging field that joins different disciplines, genetic and metabolic engineering, system biology, biochemistry, and the aim is to modify or construct from scratch new organisms that can optimize some feature that can have biological interest. The idea is that what I can create, I can understand. So if I can create something, I can understand deeply how it works. So in this, let me say, approach, we are interested in synthesizing minimal cell, that is structure, artificial structure, that having the minimal and sufficient feature, component, number of components to be considered alive. And the three features, the main features that they should be exhibit, so are self-maintenance, self-reproduction, evolvability. Of course, you can try to get this aim, this goal, starting from top. So starting from modern living cells and simplify these cells. And this is practically the Venter approach, minimizing, for instance, the genome, or starting from bottom. So starting from scratch, from simple molecules, and assembling these simple molecules inside two compartments, using both synthetic and natural molecules extracted from living beings. So we follow this approach, and our compartment are giant vesicle. So phospholipid vesicle of micro-sized dimension. And we prepare this vesicle using a well-known method that is called the droplet transfer method. So we start from oil droplets, and then we push these oil droplets across a interface that is rich from phospholipids, so they form a bilayer compartments. So in this way, inside the vesicle, you can put what you want, and it remains located inside the droplet. You can put what you want, and it remains located inside the vesicle. You can see here, for instance, some imaging from confocal microscopy, and you can see that the fluorescence probe that is inside the vesicle remains inside the vesicle. Also, you can labrate the membrane of the vesicle. They are very useful because they are big, so you can manipulate with like cell, and you can also monitor with technical analytica that you use with the cell, so microscopy or cytometry. Okay, we are interested in create energetically autonomous artificial cells. So mimicking, of course, nature. So we was inspired by biological living organisms, in particular bacteria, photosynthetic bacteria. In photosynthetic bacteria, in the cytoplasmatic membrane, there is three enzymes that can transduce light energy into chemical energy in form of ATP. So in principle, this is the three enzymes that are involved in this process, in the photosynthesis, in bacterial photosynthesis. There are two enzymes, the reaction center and the cytochrome BC1 complex, that works together, that are coupled with two shuttles, electron shuttles, and the they transduce light energy into a pH gradient across the membrane. This pH gradient is exploited by the ATP synthase for the phosphorylation of ADP, so to create ATP that supply energy for the metabolic process. So we use then two different approaches, a single compartment approach and multi-compartment approach to realize giant vesicle able to transduce light energy and chemical energy. And in the single approach, the idea is to reconstitute all the three enzymes inside the lipid membrane of a giant vesicle. In the multi-compartment approach, the idea is to extract some organelle from the bacteria that contains the three enzymes and to put these organelles inside the giant vesicle. So we start from the multi-compartment approach. This is our first result. In this case, we reconstitute the reaction center into the membrane of the giant vesicle. We use a reaction center that was labeled with the fluorescence probe moiety. And so you can see that the membrane is red because the reaction center labeled goes into the membrane. We use the droplet transfer method, so you use the micelles that contains the RSC into the inside volume, and these drive the reconstitution of RC in the membrane in a physiological order with 90% of success. So under illumination, we can observe that the pH inside to the vesicle can increase due to the fact that this protein is able to transduce the light energy into a pH gradient. And since we put inside a fluorescence probe that is sensitive to the pH, the increase of fluorescence is related to the increase of the pH. So in principle, in this case, we used an excess of electron shuttle that are used from the RC because we don't have the other enzyme, the BC1, that works is coupled and work in a photocycle. So we try to couple the second enzyme not in a membrane, but in a free solution of micelles. And we can see that these two enzymes can work together in a photocycle. And you can see here, these are practically the waves of this compound here, that is the cytochrome reduced. And these waves are due to the fact that during the red phase here, we are switch on the light, and so the RC works, and then it transforms the reduced form in the oxided form. When the switch is off, then in this case, only BC1 works, and then we restore the level of the reduced form of cytochrome. So in principle, this was interesting, but we have another idea. We can couple the BC1, not the bacterial one, but the BC1 extracted from mitochondria. So the BC1 extracted from mitochondria is much more efficient. So if we couple these two enzymes, we can obtain that we obtain the same performance, but with a concentration of the BC1 extracted from mitochondria that is 40 times lower than the bacterial version. So in principle, we can also increase the concentration of the BC1, and we can see that we can, in this case, and we can increase the efficiency of the system, because the stationary state, you can see, is always the level of the reduced form of cytochrome is higher when is higher the BCM, the BC1 concentration, because BC1 concentration produce the the reduced form of the cytochrome. So in this way, we can we find the right ratio between these two enzyme. But we stop here this kind of research, the single compartment approach, and we start to work with the multi-compartment approach, because the idea is instead to extract all the enzyme and reconstitute all the enzyme one by one in the membrane of a giant vesicle. It could be very complex because there is a problem also with the orientation in the membrane of this enzyme. We thought that was better to extract completely the apparatus from the bacteria and encapsulate this apparatus into the giant vesicle. The apparatus that we can extract is in form of chromatophore that are small nano-sized vesicle that contains all the three enzymes. We can obtain these organelles by breaking the membrane of the bacteria, the RS spheroides, by a French press step that is increasing the pressure on the solution, and then we can collect, we optimize this procedure to collect the chromatophore, and at the end we obtain these organelles. This is a version, a hypothetical model that has been realized by these authors about of a chromatophore, and the size is more or less 45 nanometers, and our organelles instead is larger. We measure this kind of dimension. This is some EM analysis. You can see that they are perfectly rounded, and there is also here you can see the ATP synthase. This is an enlargement that is orientated in the right direction. So in principle, we was able to extract these organelles, and we also try the activity of these organelles. If under irradiation, they can produce ATP in presence of ADP and phosphate. And we use the luciferase in essence assay, and we was able to find that under 3 minutes of irradiation, we get the 60% of yield of conversion of ADP in ATP. And then at the end, what we have to try to do is to encapsulate these in a giant vesicle. To be sure that this happens, we labeled the membrane with a lyophilic dye that labeled the membrane, and then we put these in the internal volume of our droplet solution. Then we created the micelles with the vesicle, sorry, with the chromatophores, and we observe a red fluorescence inside the vesicle. That means that the chromatophores are encapsulated inside the vesicle. They are homogeneously dispersed inside the vesicle. So our final experiment was to demonstrate that the ATP that is produced driven by light by our chromatophore are able to sustain the transcription of DNA. So we encapsulate inside the giant vesicle the chromatophore and a kit for the transcription of DNA with what everything is necessary for the transcription except the ATP, because the ATP should be produced by the chromatophore. We also add the acrinidine orange, that is a fluorescence dye, is a compound that when formed complex with mRNA, give a fluorescence intensity emission. So this is the result of our experiments. So in principle, you can see that in many giant vesicles, you can observe the formation and the increase of a fluorescence due to the formation of RNA and the formation of the complex with the acrinidine orange. And then this is just the last experiment. Of course, we did a statistical analysis about this results, and we observed that, okay, the giant vesicles are very dispersed in size, and if you analyze the synthesis, the intensity of the fluorescence, we can observe that the Gaussian distribution can explain this dispersion, but there are a certain number of giant vesicles that exhibit a very large emission. So that is not described by the Gaussian distribution. This is something that is a crowding effect. Maybe Luigi mentioned this effect yesterday, I don't know, but is always observed when you are working with the small compartment. And in principle, and we also was able to measure the rate of the ATP production inside our groove. Okay, so I'm practically finished. Okay, and as a conclusion, we was able to realize the artificial giant vesicles that are able to transduce light energy into chemical energy. In the case of the multi-compartment, the single compartment approach, this transduction is in terms of pH gradient. In the case of multi-compartment, this means the conversion of ADP in ATP. And so the multi-compartment approach, of course, is much more efficient, but in this line we can use also other organelles that can be used. For instance, now we are working on the chloroplast. Okay, let me just thank the people that are involved in this research. Paolo Albanese and Emiliano Altamura that are deeply involved experimentally, Pasquale Stano, Massimo Trotta, Francesco Milano, and Peter Valde that are collaborate with me, some sponsor, and also I would like to thank the academic of American National Academic, because they give us the Cozzarelli Prize for as the best paper in the 2021 about this system. Okay, and thank you for the kind attention.